All right, good morning. Today we're going to be looking at the coral reefs, abiotic and biotic factors gizmo. So we're going to be looking at the introduction now, and then we'll talk briefly about activities A and B. So for the introduction warm-up, what we're going to look at here is some of the basic information that they're trying to get across. We have the vocabulary up here: consumer, coral, coral reef, filter feeder, food chain, food web, grazer, nutrients, plankton, predator, producer, sediment. Now, all of these can be fine to look them up. Right here on the lesson info, you can see the vocabulary sheet right there. You can find all of the terms for this gizmo all right there. Prior knowledge. So what we're going to be looking at here is they're asking to look at the graph here on the side. They're asking, what does it show? So it's labeled the percentage of coral reef in the Caribbean reefs, 1977 to 2012. Question 2 is asking, why do you think the coral reefs have declined? So why is it that you think this might have declined? The warm-up, and here we're looking at how you are able to manipulate this gizmo. So it says click advanced year 10 times. Right here, advanced year. So we've gone through 10 years. Do you see any major changes? Is anything major changed? Does it look like much has changed? Still have pretty much the same organisms. We go through and look at the summary to find more. Select the data tab. So we'll look at data, and it's asking to check these 4 things. It's asking to check staghorn coral, star coral, sponges, and algae. Those are all selected. What do you notice about the populations? Well, you'll notice the populations pretty steady, except for sponges, which have slightly increased. On the right side of the gizmo, you'll see summary and a healthy reef. What are the values of the nutrient load, water, clarity, coral cover, and the total number of fish species? So you'll go ahead and look at this. We'll look at summary, and you can put down this information there. All right, so activity A. This one is looking at describing the different organisms. So we'll go back to conditions, back to the coral reef, click restart, and we're all ready to go on this. So we'll restart back to year 1. Now, what it's asking us to do is to go through and give a brief description of the different organisms. So it gives you a picture so you can find them. So we'll start here. Staghorn coral, fastest growing, most delicate Caribbean coral. It obtains its energy from photosynthetic symbiotic algae. So it gets all of its nutrition from an algae that does photosynthesis. So I'll put here staghorn coral, what it eats or how it obtains energy. It gets its energy from photosynthetic symbiotic algae. And then you'll do the same thing with each of the other organisms here. You'll click on it, put its name, write down what it eats, so on. Activity 2, how do you think corals would be affected by cloudy, muddy water? So if it were suddenly to become cloudy, how do you think that would affect all the coral in there? Classify. Which of the reef organisms in this gizmo are producers? So you'd look for the things in here that are producers. Now you'll know that there are producers because a producer has something to do with algae, something to do with photosynthesis. Consumers in the reef can be classified as filter feeders. They eat plankton by filtering water. Grazers, organisms that feed on organisms that don't move. And finally, predators, organisms that eat other animals. Go ahead and give an example of a filter feeder, a grazer, and a predator here. Final question for activity A is our big point for this. What we're looking at is food chains. You need to create 3 food chains where you start with a producer, something that gets its energy from photosynthesis from the sun. You'll then go through and create something that eats off of the producers, and so on all the way through. You should be able to get at least 4 for each of these, 4 parts in the food chain. A producer, something that eats from the producer, something that eats that, pretty easily. Excuse me. Activity B is looking at invasive species. Now, this is where humans are really coming into effect and changing ecosystems. We're going to be looking at the red lionfish. So, the red lionfish is a native of the Pacific Ocean. It was first observed in the Atlantic Ocean in 1985. Red lionfish are voracious predators and are well protected by their toxic spines. Today, they have spread throughout the Caribbean. So, the red lionfish is a creature that does not belong in the Caribbean. And what's worse, it is actually a creature that doesn't have any predators in the Caribbean. So it can just eat whatever it wants. So what you're going to be looking at in this one, it's going to ask you, what do you think is going to happen? You'll notice here, we have the steps of an experiment. You're going to predict what's going to happen to the reef when you put in the lionfish. You'll observe it. Here's the experiment going 20 years. We'll look at an observation again. You're going to predict how you think the reef will react to a lionfish invasion if there's no urchins, and so on. You're going to experiment again, predicting what's supposed to happen with each of the different parts there. Which brings us to our last portion of this gizmo. And this is the big one that we're going to be looking at, the extend your thinking. You're going to be creating your own experiment. You're going to test your own thing, okay? So you can test how different storms, you can test how the temperature, you can test pH, you can look at the different land uses, logging, dumping sewage, agriculture nearby. You can look at fishing. You can test and see how more fishing or less fishing will affect it. You can check and see how different diseases will come in and affect the ecosystem. You can even look at doing another one with the lionfish if you so choose. So you're going to create your question and your hypothesis. Your hypothesis is going to be the answer to your question. In other words, how is fishing going to affect the ecosystem? Then you'll put your design. You're going to design your experiment. What are you going to be testing for? How are you going to test? How many years is this going to go through? All of those different things that you can change in there. Next, you have your results. What did your results show? What information was shown by your experiment? And finally, your conclusion. Was your hypothesis, was your answer to your question correct? Okay, that is the gizmo. Make sure that you get it done. We have a lot of other experiments and different things we're going to be working on for the next couple of weeks. Have a good day.